Hey, I'm Abbie, and I'm a forensics major at Arizona State University.
Hi, I'm Simona, and I'm also a forensics major at ASU.
And my name is McKenna, and I am a biology major with an emphasis in forensic science.
Our learning objectives for today are to be able to describe the prominent discoveries made about CRISPR, its origins, and how the CRISPR process works in general, as well as how CRISPR technology has the potential to advance the field of genetic engineering, and the ethical implications that correspond with the use of CRISPR-Cas9 in gene editing. When we think of bacteria, we usually think of how dangerous they are to us. However, both bacteria and ourselves share a common enemy: viruses. In fact, it is a dangerous world out there for bacteria. Viruses outnumber them by about 10 to 1, and viruses are believed to kill about half of the world's bacteria every 2 days. When a virus latches onto a bacteria, it only has a few moments to defuse the situation. One method that about 1/3 of bacteria and 90% of archaea use in order to survive against viruses' attempt on their life is an immune system called CRISPR.
CRISPR is an abbreviation for Clustered Regulatory Interspaced Short Palindromic Repeats. The palindromic pieces of DNA, or sequences of genomes which are read the same way forwards and backwards, can be found in the genomes of several types of bacteria and archaea. These types of sequences come from breaks or inversions in chromosomes, and are interesting because some restriction enzymes will recognize them and cut them out. Despite many scientists having known about the palindromic repeats in the genomes of several organisms for years, it was not until 1992 that Francisco Mojica from the University of Alicante in Spain found a common set of features shared between the repeated sequences. He would later coin this common set of features as CRISPR. Despite other scientists deterring him from his research, Mojica's determination to uncover what these repeats were led to the modern discoveries made by other scientists that reveal CRISPR's potential. Here's a quick overview of the discoveries made following Francisco Mojica's work that have led to our understanding of CRISPR-Cas9 today. In 2005, Alexandre Bulletin worked for the French National Institute for Agricultural Research and discovered a novel Cas protein, now known as the Cas9 protein. It was not until 2006 that Eugene Koonin hypothesized CRISPR as a bacterial immune system, and it took another year for Philippe Hovarth to prove CRISPR was a bacterial immune system. In 2008, John van der Oos from a university in Netherlands discovered that RNA was responsible for guiding the Cas proteins to the target DNA. Later that year, Luciano Marafini and Eric Sonheimer at Northwestern University proved that DNA was the molecule being targeted. Sylvain Manoir from the University of Laval in Canada demonstrated that the CRISPR-Cas9 complex creates breaks in the double-stranded DNA, and that the Cas9 protein specifically is responsible for cleaving or breaking that targeted DNA. In 2011, Virginia Signis from Vilnius University in Lithuania revealed that CRISPR systems could function in other species as well. All of these discoveries led up to Emmanuel Charpentier, director at the Max Planck Institute for Infection Biology, and Jennifer Doudna, a molecular biologist at UC Berkeley, 2012 discovery of CRISPR-Cas9 and its potential to edit the various traits that are present on a genome. But before we get too far into the potential of CRISPR, first let's take a look at its origins. The exact origin of CRISPR-Cas9 immunity is still unknown. However, it was predicted by Eugene Koonin to evolve from transposons, which are little pieces of genes that are able to hop to different areas of the genome, and make up about 44% of our own genome. Koonin and his colleagues based this prediction on his discovery of the Cas1 protein. This protein is involved in placing spacers into the genome, which parallels the function of transposons, which is why they're called casposons. Now let's take a look at how CRISPR and the Cas9 protein work together. When a virus binds to a bacteria cell, it acts as a syringe to shoot its DNA, or in some cases its RNA, into the bacterial cytoplasm. The CRISPR system will then capture the palindromic sequences of the evading viral genetic information and use it to create spacers. These spacers are short DNA sequences from a previous viral attack on the bacteria that are stored as memory in the bacteria. They serve as a genetic record of all viruses the bacteria has come in contact with. The bacteria will then transcribe the DNA spacers into RNA, which will be exact replicas of the viral DNA that the DNA spacers were made from. Next, the RNA will bind to the Cas9 protein and form a complex. This RNA-Cas9 complex will filter through all of the viral DNA, targeting the viral palindromic sequences until it finds a match. When a match is found, the Cas9 cleaver acts like a scissors and will cut up the viral DNA. By targeting the viral DNA through an RNA match, the bacteria is able to recognize their invader, diffuse the situation, and get rid of viral threats. Additionally, should the bacteria or any other cell be attacked again by the same type of virus, it will be able to defeat the threat again as long as it retains the DNA spacer located on the CRISPR site.
So why should we care that both archaea and bacteria have evolved to defeat viral threats? Well, what is extraordinary about this process is that the CRISPR-Cas9 protein complex can be programmed. We can insert exactly which sequences we want to be broken. This will allow scientists to target and study particular DNA sequences in genomes. And doing what scientists always do to discover how something works, we will be able to break or disrupt a gene and learn what that gene does. Additionally, plant and animal cells already have ways of reconnecting broken fragments of double-stranded DNA. This can be done by binding the two broken ends, also known as disruption, or by incorporating new genetic information into the broken sequences if it fits. So by being able to program the CRISPR-Cas9 complex, we will be able to combat diseases such as cystic fibrosis, autism, sickle cell anemia, neurofibromatosis, and multiple types of cancer, including breast, colon, and skin cancer, and many other diseases that are a result of genetic abnormalities, simply by cutting out the abnormal gene and replacing it with healthy sequences. In fact, there are already studies being planned that will incorporate CRISPR-Cas9 into cancer therapies. One particular study plans on removing the T cells, a type of immune cell, from 18 patients with different types of cancer, and then inputting 3 CRISPR edits into them. One of the CRISPR edits will be a gene that codes for a protein that is able to detect cancer cells and instruct T cells to destroy them. The second edit is meant to remove the patient's own T cells, as they might interfere with the previous process. Finally, the third edit will remove the gene that works to identify T cells and prevent cancer cells from being able to disable them. Combating genetic diseases is unlikely to be the only thing these advancements in genetic engineering have the potential of leading to. There is also the possibility that CRISPR will be used to select genetically favorable traits. If we can figure out how to use it successfully, it could one day be used to change virtually any trait or characteristic that is present but unfavorable in humans. Already, we have seen evidence of this occurring in Jennifer Doudna's lab. She was able to change the coat colors of mice with the help of CRISPR. One's hair color, height, intelligence, skin color, eye color, and/or even their athletic capabilities could all become purchasable traits within the next few decades. Changing the traits of an individual is only a fraction of the capability that CRISPR technology has. In fact, CRISPR technology could be used to edit an entire population's genes, also known as a process called gene drive. This could put an end to disease-carrying mosquitoes, eliminate invasive plants, or get rid of herbicide-resistant pigweed plants. However, the consequences of being able to eliminate certain traits from an entire population has unknown effects on the ecosystem and the species residing in them.
CRISPR can be, as aforementioned, effectively used in humans and other animals. It is also capable of being replicated into Dr. Sweat's favorite topic, plant life. By replicating CRISPR into agriculture, we could be able to produce strains of cereal grains and root vegetables that are able to withstand viral invasion, essentially creating a way to combat problems like world hunger. Currently, CRISPR is being used to make disease-resistant wheat and rice, as well as vitamin-enriched sweet oranges. This new improvement would make it harder for people to track products that have been engineered, especially because in the United States, not all crops made by genome editing would be required to be regulated by the U.S. Department of Agriculture. However, it is with this advancement a larger percentage of food can be produced and maintained. So introducing this new form of virally immune plants to other countries, especially those with high levels of poverty, can make food more accessible to the hungry. As a result of its recent discovered impacts in genetic engineering, CRISPR-Cas9 has gained a lot of attention from various researchers. A study currently going on in China illustrates how close we are to being able to implement this technology. A trial run by Dr. Yao Lu and Dr. Shenglin Ma of Sichuan University is looking at the implications of CRISPR on non-small cell lung cancer. Their research aims to take the protein that causes sudden cell death and remove it from the gene sequence with CRISPR. They will then culture it ex vivo and reintroduce it into people with the disease. They think this could be a way to cure lung cancer without ever subjecting patients to chemoradiation. Along with the use of CRISPR becoming more widespread, so does the fear that the genes will be used to enhance or eliminate traits for non-medical reasons. It could also lead to genetic classism, because not all people have access to this new form of gene editing. Many fear, too, that there can be unintended consequences for gene editing spanning over several generations of people, past the initial introduction of the gene into its new hosts. In several countries around the world, there are measures in place to either restrict or outlaw the use of CRISPR proteins for genetic modification. This is in response to, generally, the use of human embryos in genetic experimentation with CRISPR, and has little to do with the general use of CRISPR for other organisms. Researchers in Canada brought two important legislative issues to the table, saying that concerns are coming from the questioned acceptability of human embryonic stem cells, which is generally deemed acceptable, and other reproductive cloning. Though China and other countries have bans and restrictions on gene alteration, many people literally do not give a fox and go along to do that shit anyway. This is why we see some of the first successful human embryonic work coming out of China in 2015. In these experiments performed by Zhenzhu Huang at Sun Yat-sen University, Huang found that they were more often than not very off in their projections. In one test with 86 human embryos, only 71 survived and 54 were genetically tested. There were only 28 that had been successfully spliced. The scientists found this percentage was too low to warrant further testing, so they labeled the gene too immature for the time being. There were also a number of off-target mutations observed by the team, and this was just what many people were concerned about happening. There has not yet been any successfully created babies with the CRISPR gene, nor has there been any astounding resolve to make it happen right away, because the technology has proved to not yet be completely ready. Some of the first babies who might be born with CRISPR will be in countries with a high rate of in vitro fertilization, like Japan or India. The CRISPR sequence has the potential to do so much good in the world, but it also has the potential to do a lot of harm. Whether these genes will ultimately be used for the right reasons will be up to the scientific community as a whole.